Karl and his team here at John Redcliffe have been focusing their research on understanding the role of mitochondria in health and disease, working across a range of conditions, including ME. This work is much needed in order to help us find some clues to the causes and hopefully some of the cures for ME and related conditions such as long COVID. We are thankful for his work in this area and the ongoing relationship with Omega. I know they have lots to tell us about their exciting work, but I'd just like to take this opportunity, being a beacon of hope in what's been a sort of dark and stormy time for many people. So thank you. Thank you.
Thank you, Anne. Welcome everybody to the JR. This is one of the better rooms. We have a bit of leaks for other parts of the building, so you're in the best place. So I'm going to introduce you to my group. We started about 6 years ago, I think, roughly in this space. And things have come on, not just with our group, but I think globally, there's a lot more research going on in ME/CFS, and long COVID has made a bit of a difference, you know, because people are more aware, I think, now that these things are real. So we're going to kick off with Inga, one of the PhD students from my lab. She's going to tell you about an exciting new study which is about to start. So I hope you're all listening online, because we need people to volunteer to come forward. So Inga's going to give you a bit of an overview of that.
So hello, I'm Inga. I'm obstetrician and gynecologist, and I started quite a few years ago with, sorry. Yeah, okay. Do I need to be more loud? Okay, sorry. So I'm obstetrician and gynecologist by trade, and I started many years ago by studying infections and effect of infection development of the chronic conditions in obstetrics and gynecology. So I'm presenting my study, which is causal mechanism in fatigue. So presenting my team. So principal investigator is Professor Karl Morten. I'm primary researcher for this project and the supervision of a wonderful Dr. Beata Godlewska, Dr. Jenny Mons, and Dr. Tiffany Lynch. So why us? Because between us, we are a team of scientists and clinician scientists. We have expertise in myalgic encephalomyelitis, chronic fatigue, and long COVID, and also in mitochondrial disorders. So the problem we would like to address is that we all know that fatigue is a common in many illnesses, and it's one of the features in myalgic encephalomyelitis, chronic fatigue, and long COVID. And as we all know, many factors are involved in development of myalgic encephalomyelitis and chronic fatigue and long COVID. And what we would like to answer is there a common cause of fatigue in different illnesses? So our aim is to find out, can bacterial imbalances cause fatigue? How do immune, infective, and mind-body factors modulate fatigue in different disorders? And how such knowledge could be used? So we would like to introduce this study, and you can take a part if you're suffering from myalgic encephalomyelitis, chronic fatigue syndrome, or long COVID after viral illness, or you're well relative of a participant who suffers from myalgic encephalomyelitis, chronic fatigue syndrome, or long COVID. You're 18 and 55 years old.
Yeah, lots of old people. They're the ones who are going to be struggling.
I can answer the questions a bit later. Sorry. And we wouldn't be able to include you at this stage. This is studies, pilot studies. So we're only recruiting at the moment 20 people from each group for each group. So it's 20 people from a group of long COVID, 20 for myalgic encephalomyelitis, chronic fatigue syndrome, and 20 participants. So we wouldn't be able to include you if you are receiving regular ongoing treatment for other problems. If you used antibiotics, antifungal, and antiviral medication in the past 3 months and had recent treatment for cancer, and also if you're struggling with weight issues. So what will happen during the eligibility assessment if you decided to take a part in the study? So we would like you to attend a video interview where we're going to be discussing inclusion and exclusion criteria, I will answer your questions about the study after sending you all the documents. So we will record verbal consent. We'll ask you to fill in questionnaires to assess your mood, fatigue, sleep, gut health, and diet. So when we invite you to take a part in our study, before the study you'll receive a box with containers and instructions how to collect the samples. So to the study you'll bring 3 samples of one of the hair, urine, and saliva, and we'll ask you to send a stool sample and complete 7 days diaries of the diet and sleep. The study visit itself is going to be one study, and it will last 90 minutes. So for that, you can choose a site. So you can either be visited at home, or you can come to us to this building, or another site is going to be not very far from here, but more easily accessible by car, and there is no problems with parking as such. It's going to be in Brookes University, which is not very far from here. So we would like you to listen to the study description, ask questions, sign a consent form, and fill in 2 short questionnaires, and that is about your current health, because quite often happens that from the moment we had a first interview and actually conducting the study might be a few weeks. So we would like to assess your current state of health and ability to do activities. So during the actual visit, we would like you to wear a watch to look at your heart rate, blood pressure, level of oxygenation, and also EEG cup. We would like you to do an attention test, memory test, and also copy a little text, like a little poem for us. And at the end, we would like you to provide a blood sample. So after collecting all of this, we would like you to, we would give you a watch either before the study, before you come to the study, to collect all the necessary information like blood pressure, heart rate, and oxygenation, and possibly sleep patterns and things like that. And all this information will be collected in order to test our hypothesis, which is on the screen for you. So we think that there are particular cell wall deficient bacteria, which you can see on the right, and which I have been studying a long time ago, and which can potentially cause autoimmunity and can cause the gut dysbiosis, which is disbalance of the microbes, pathogens in the gut, which might potentially cause leakiness of the barriers. So therefore, these pathogens can hitch a ride inside of the cells and cross the barriers and get into the brain and cause the brain changes and cause the symptoms. So of course, it will affect things like gut and will also contribute to a reinfection. So this is our study design. So there are some markers which we'll be collecting in order to test this hypothesis. And I would like to say thank you to our private donors for your very generous donations for us to our study, to my supervisors, Professor Karl Morten, Dr. Gadlewska, Dr. Miles, and Lodge, and Action for Me Foundation, to my Future Leaders Network for their contributions, and to my wonderful husband, Dr. John Williams, who is just at the back of with his help with this study, and Project Angel, or donor and source of inspiration in exploration of human abilities, Ola, who is there on the screen. She's a free diver. And thank you very much to all my my family. We can see my wonderful children, and to my alma mater in Kazan, in Milan, in New Zealand, and in Oxford. Thank you.
I think there's a few questions come in, so I think we'll take some questions. I think there's maybe a few questions from the audience as well. Right, okay. So, any questions from in the audience at the moment? Yeah.
Could you say a bit more about how you use the information you're going to gather to answer the questions that you said at the beginning?
So it's not very easy, but we will be using very advanced methods of statistics, and it will be done with help of our people in a statistical department, as well as with outside advisors as well. So it hasn't been done yet in myalgic encephalomyelitis. It's been done in the medicine in general, but these methods are widely used in finances and sociology.
So they're trying to look for move from associations to more causal factors. You know, we've all got problems with POTS and blood pressure changes. Trying to link those to things that we see and maybe either biological markers that we can measure to see whether we can get a hand, because there's so many things that are different. We don't really know what's driving this illness. Let's just try and see whether we can get a hand on them. Some of the new methods that Inga's going to apply might help us do that with just a single sample rather than longitudinal samples, which take years to do. We've got a limited budget, limited numbers. There's only going to be 20 in each group. So it's just very much a pilot study, a feasibility study. Can we do it? Can we do it here in the hospital? And can we recruit people? I think somebody has a few comments online. So by weight issues, do you mean eating disorders or weight problems associated with ME? What adjustments are you making to ensure people with severe ME are included?
So first of all, I will answer the last question. The adjustments to ensure that people with severe ME included, we very much like you to take a part. Please approach us. We would like to travel to you. We will be wearing masks and doing everything to protect you while we're visiting you. So by weight issues, we are talking about weight issues which are caused as there is before. What you did you have? Did you experience problems with weight issues before you got ME? And of course, we all know that diabetes and conditions like that might be contributing to the problems which might potentially disturb the signal of the data which we'll be collecting. So therefore, I'm very sorry at this stage we wouldn't be able to include people who are very overweight, but we would very much like you to be involved in a future study which we hope going to happen in the future.
How far would you travel to see people with severe ME?
Up to 1 hour.
Yeah, we've got some travel budget, so we can pay for people to come here.
We have got 10 years old, so to collect from school.
Yeah, and the timing. So yeah. It is very much a pilot. We'd like to include everybody, but what we found from, so Jamie might mention this in a minute, we've done lots of studies of blood, chemicals in blood, and we've shown that just 2 BMI increments, not somebody that's obese, have made a big difference to some of our data. So we want to try and take what is a very heterogeneous condition and try and make it as narrow as we can. So that's why the age, 55, I think there's changes in the microbiome as we get to sort of 60. So we're trying to keep people as close so we can get some sensible answers rather than confounders that come in and might not be what we want. So this is a pilot, and there will be bigger studies. It would be great to have a severe group because we've seen big differences in the severe groups in other work. I'll talk about that in a minute. Catchment area nationwide, probably not. Probably, as you say, within an hour of Oxford, if people can get here. We have got some budget, but we need to spend a lot of that budget on the experiments, really, so we can't really afford that. Comorbidities will be excluded.
So I would, if you're interested, please contact us, and we will. There's an advertisement on Morten's group page, and as well as we probably will send you email address, which you need to use to contact us, and it will be a list of comorbidities which we wouldn't be able to include at this stage. It is very pity, but it's very much to have an idea of what we're looking for at this stage.
Yeah, if we go too broad, we might not get any useful information, so that's the reason why we're trying to keep it as best we can. I knew there'd be loads of questions, as always the way we make it. Can participants view the results of various tests they complete, bloods?
So at the end of the study, before we're going to be publishing everything, we will be issuing the very document which will tell you all about what we found. So very much like you to ask the questions. We would be able to answer questions about this study. So please ask questions.
Sarah says, further to Helen Baxter's question about access for people with severe ME, is your study designed to prevent triggering ME? Can those severely affected pace their involvement over weeks, blood test over 1 week, questionnaire another?
Yes. So the questionnaires are going to be done well before the study, the main lot, and you will be able to answer your questions. So we would like you to use as much help as possible. But if you're recording the answers of using your helper, please ask them to record what you say, not what they think they thought you said. So, and of course, when we're going to be doing this study, once we're visiting you, we would do a very short questionnaire, and we'll take the blood at the same time, just to see what is happening as a result of the study at the moment.
Thank you for inviting us to your presentation. I think we'll stop these questions, otherwise we're not going to get to the rest of the questions.
Just one. Michael's one.
Michael's one. I mean data on individual participant results, not just the whole cohort. I think you were going to do that. You're going to give individual results, or
we're not allowed to.
Yeah, so just the whole cohort.
Yeah, it's not. I don't want to give you the answers, but your query, we're very much respectful.
We don't know what they mean. It's not like going to the doctors to do a test. We can't really give you any information, so we'll tell you the whole study. Because we published our own research, and everybody wants us to do this test. It's not really a test yet, so we need to just be clear what we're doing. So. Okay.
Okay.
So Edie, shout.
Okay. All right, everyone. I'm really happy to be with you today. It's actually my birthday today, so it's no other way to spend it. Thank you. Because it's a lot of hard work in the lab, and the point is we want to make your life better. So it's really exciting to share what research we're doing in the lab with you guys. So my DPhil project is on identifying factors in the blood. Oh, sorry, I forgot to mention. So I'm Edie. I'm a third-year DPhil in Karl's lab. My project is on identifying factors in the blood which could impact on the cellular energetic. That could be like a cause for ME, and then also trying to develop a diagnostic test. So I'm going to share some data later on. I'd prefer if you guys just kept it to yourself and don't share it. Okay, so my supervisor is obviously Karl, and then my other supervisor, secondary supervisor, is Wei Huang. He is in charge of this Raman machine, a light microscopy that's going to help us develop the diagnostic to find out a cause or factor, and then Tiffany. So introduction. I think I included this, but I think most of you already know what ME is. I'll just go over it generally. Issue is no biomarker, no diagnostic test, no cure, and very limited treatment. Most patients aren't diagnosed. I think long COVID has helped make it better, but it's, obviously sort of awful illness. And just go over some previous research that's in literature, what other people have been doing. So this also kind of set the background as to why I am doing my current research. Presti's group, they have found there's mitochondrial dysfunction. So I think, as Karl has mentioned earlier, and Inga, there's issues. So mitochondria is like the powerhouse of the cell. So if there's the problems with the energy producers, then that could be a cause for the fatigue and ME. So. But they also did find some biological issues with it. So there's issues in mitochondria. There's also impaired issues with pyruvate dehydrogenase dysfunction. So there's some kind of metabolic issue there. And then these other two papers, they're just examples of what's showing there's dysfunctions in the endothelium. So there's changes in nitric oxide production. So that's to do with endothelial function, how it vasodilates, and then there's also so a group, they put the serum from post COVID-19 patients, syndrome patients with or without ME, and then they put it onto endothelial cells, and they saw there was an effect. So that's just a kind of background of where my research would kind of go in to fill in the gaps and expand on it more. So our approach is that this is the Raman microscope. So when I mentioned one of my supervisors has a microscope. This is a thing. So this is not ours because I don't have an image, but the idea is you have like, it's like a very powerful laser, and then it shines onto the samples that you see here. And then, so this would be like different kind of cells you can put on, and then they'll generate an image. I'll go into it a little bit more here. But so yeah, so that's my aim is to first identify a factor and then also do a diagnostic test. So the factors in the blood that could be a cause, maybe something in the plasma, serum, certain viruses. I think there's a lot of research that's shown on like herpesviruses, molecules, and then for us, we'll analyze the mitochondria. And then for the diagnostic part, so some of this is going to be a little mixed because with the technology, we're going to use the Raman, but for the diagnostics, it's going to be the Raman microscope, but also this other new system called the ILAF. So this Raman is it's been around for a while. The person that developed it, Raman, he actually won the Nobel Prize a long time ago. So the ILAF, but it's very expensive, and it takes a long time to process the samples. So the ILAF could be faster and cheaper. It's a newer technology. It's based on a startup. So that could be like an alternative. So the idea is I'll develop them simultaneously and see which one is better, which one is cheaper. So I'll just go over the Raman a little bit because it'll help you understand like what I'm doing with the samples. So this is the Raman, like the image shown before. So the idea is like you have this, it's a molecule. So in the body, there's like, we're made of like little molecules, right? So for example, and then these molecules, they vibrate. They're just constantly moving around. So, and then when you hit it with a laser, like a powerful laser, most of the light will bounce up at the same wavelength, meaning the same energy. But then because they're vibrating, sometimes in a very, very small percentage, like 0.000 many 0.01% of the light, it'll bounce off with a different kind of scatter because it either gained or lost energy due to these vibrations. So that's called the Raman scatter. And then you can graph it like here. So this is called the Raman shift, some intensity. So you can see, so like the idea is every single molecule has a different, it's like a very unique like spectral fingerprint. So like just like a person's fingerprint, it should help you identify, fingerprint can help identify a person. This will help you identify a molecule. So we want to do that with the like the serum, for example, because maybe some of these pixels tell you like maybe this is like a lipid or something's a structure in the cell. Some parts could be like heavy metal, could be like but it's not really. So that's kind of the idea with the Raman. So with the Raman, I will use this to look at this. So I will put the serum of like healthy NME patients and put it on to the HUVEC cells. So that's going to be our model cells HUVECs are like a model endothelial cell, which will help us understand what the effects are on the endothelium. So endothelium is like the blood vessels and things like that, capillaries. And so then I'll use that to see if there's any kind of like structural changes or any kind of like metabolic changes in the endothelium. And then this part 2 is to use this to make a diagnostic test. I think I said that the other way. All right, so and then previously in the lab, we have, so this is a paper that was published in the lab earlier this year. Essentially, I think Karl will go into this in more detail, but essentially what happens is you use, so you have like 100% of the patients, right? And then you create the samples, and then you have your data, and then that's 100% of the data, and then you take 80% of it to build a model of categorical models, and then you put in the 20% to see like whether or not this model can actually predict a person is healthy or have ME. So based on this, and then some like really advanced statistical methods, and then various different statistical methods, and there's a combined method which can give an 84% accuracy. The issue is using PBMCs. PBMCs are different cell types. It's kind of tricky to obtain. So we want to improve the accuracy, and we want to improve and make it cheaper and easier and faster. So this is a little pilot that I've done in the lab, which is, so these are, imagine these are little cubic cells. So like they're obtained from the umbilical cord, the endothelium from the umbilical cord. So they help us model what the endothelium would, like what the capillaries. And then, so I grow them essentially, and then I add the treatment. So this is where so Inga's collecting serum. This is where the serum comes in. So then I'll put in serum, healthy, and ME, or no serum, just as a control, and then see what the effects. And then in the end, I put it on a slide and then look at it under the microscope. So there, I'm going to be doing single cell Raman microscope. So it means I will be looking at individual cells. You kind of, so you zap it, and then you see what the fingerprint looks like. So yeah, so then the end result is that you get like a fingerprint like this, and you measure it like multiple times, and then through some like data processing that you see here, it's not important that you understand what this is. It's just showing that it's a lot of data processing, and we're going to be using some like statistical methods. I think Jamie might go over later. Yeah. So the idea is like over here, for example, this is one of the methods. So it's LD. It's one of the stats models. And then you can have like all these little circles, and then the idea is like maybe they will show you the different categories. Now there's going to be a lot more processing and statistical tests that needs to be done before this could be like a good model, but this is kind of the idea. And this will help us identify if there's any kind of differences in peaks. Yeah, so as I mentioned earlier, to find so that was like the diagnostic, but to find a causal factor, like some of these are like, this could be like glycogen, this could be like, there's like different things, like lipids are here, like there's different peaks that correspond to stuff. So then you can have a stronger peak maybe, because there's going to have a stronger peak somewhere, a weaker peak elsewhere. The idea is if, say, MU serum produces a stronger peak somewhere, that means there's like a change, right? So that's something that we're interested in, and I'm trying to figure out. Yeah, and then also, so how are we going to study the mitochondria? So the mitochondria, again, we'll still put the serum onto the cubics, the endothelial cells, and then isolate the mitochondria, which we're able to do in the lab, and then you can put them onto the cells. So they would be alive or dead, fixed, and then you Raman them. And again, you can use that to identify the peaks to see if there's any changes in the mitochondria that'll help you us pinpoint what is going on with the mitochondria. And among like other assays we can do, like oxygen consumption with the mitochondria, but that's just one of them. And then the next step is one of our collaborators in Canada, he has this massage cuff. I don't know if you heard of it. So you put on this, so because obviously exercising is not good. So then he has this like massage cuff where it's literally, you put it on and then it gives you massages, but then in a ME patient that would introduce PEM. So this should be better than in like a experiment that would be better for them if they were exercising, right? So, but we're interested in why, like why does it introduce PEM? But also like if you collect the serum before and after someone used the cuff, then you can maybe see if these little factors that we're trying to pinpoint, like is that also changing like based on the serum from this induced PEM. So that would be quite interesting. So we do that with cubic cells and also the mitochondria. And yeah, that's thank you for listening. And also thank you to yeah, the funding is from, there's also funding from the ICANN and Meme Association, so I'd like to acknowledge them. So yeah, thank you.
Okay. Maybe there are a couple of questions, perhaps, from the, or lots of questions.
Oh, okay. Where did this start?
Any questions from the room? No, I think we're all right.
No questions? Yeah.
Oh, yeah, one question at the front.
I want to check either. So you take the blood cells from the test from us and
Yeah.
put it on the HUVEC things are generic. They're not. They're from somewhere else.
Yeah. So like for example, like Inga's going to collect the blood. So we would collect the blood from you, and then we would kind of treat it, process it so that we get the serum. The serum is like the clear part of the blood, so no red blood cells. And then so we're using that because there might be like a factor that's in the serum, and then like just taking out all like things like red blood cells that could be it could be interfering with like the test. And then putting it onto so you would grow like HUVEC cells, so they are endothelial cells, and so we previously have done things with like cancer cells, but obviously that's not representative of immune patients, right? So we want to use primary cells, which means they're taken from they're just cells taken from like the umbilical cord.
From someone else.
From me, yes. From me, like from yes. But so the idea is because obviously you don't want to like you want to study this, be able to study this in the lab. So you want to have like a model cell where you can like grow in a petri dish and then do like various different tests with it. And then so that's why I'll be using the endothelial cells and then putting the serum on and then seeing what it does to the endothelial cells in the lab. And then doing different measures like what's the respiration, what is it doing? Just trying to understand what is it doing, and then what's causing the pathology.
Keep going. Is it the HUVEC that they then put in the Raman?
Yeah. So I would take the HUVECs, and then I have them as like single cells, and then I would look at them one by one. It's a lot of manual labor, but and then you kind of analyze all the data. Yeah.
I suppose. So this is to look at the serum on a model, but we've already looked at the PBMCs, the patient samples, and I'll talk about that in a minute. So we already know what the profiles look like in patients. So we want to see what the serum does. Yeah. Does it look similar in the HUVECs or completely different from what we see in the patients? So that's one comparison, but also comparing the healthy controls and the ME patients, and also the multiple sclerosis patients in another study. So it's looking for the factors, but also the diagnostic probably might be more the serum in this experiment than the actual HUVECs. So I think Carol?
With the massage thing, the massage cuff to induce this PM, will you put the massage cuff on people who don't have ME to see so you can compare the effect?
Yeah, so like usually if you do like a control, that'd be the control you would collect so usually you do like before and after. Maybe.
I'm not.
It's amazing that it actually does anything.
I know.
Like that.
They find out that it.
Well, they don't. It's not definitely cut and dry. They look at microRNA changes and they seem similar, but no one's really looked at it in much detail. So they're quite keen to give us one.
Yeah.
Hopefully we can. Okay. Okay.
There, right? Yeah.
Oh, sorry.
Used in diagnostics at the moment, or is this a pioneering technology at the same time as well? Like, is it used more in analytics than diagnostics, or is it like a commonly used thing?
Yes. Well, the idea is we want to develop a diagnostic test. The paper that was published, so that would be like that. The idea is that would be developed as a diagnostic test, but we need to make it better to use on like in an actual like patient setting because. Like, for example, like we need to improve the accuracy, like 84% is quite good, but we need to improve that. But also like before this was looking at, so we took the blood and you take these cells out. They're called PBMCs. They're kind of like immune cell. It's basically a very like long and expensive process. You wouldn't be able to use that on like a regular testing. So it's trying to find different methods to use that method, but also trying to improve it. Yeah.
Just I'll mention this a bit. So it's been used in cancer. It's starting to come into the cancer space. So, and I'll say a bit about that. So similar, not single cells, but maybe other things. So I'll mention that. Yeah. It's very new to biology. It's been used to look at paint and fake art and things like that.
They use it. We used to use it to like identify like microplastic. You can like zap it and then you see what kind of plastic it is, like polyethylene, polypropylene. So, yeah.
So hi everyone, I'm Jamie Strong, and for a while I've been working with Karl and the team on our metabolomics projects, where we've collaborated mostly with our group in Poland, Slavomir, and
James,
Yeah, James McCulloch, based at Oxford, on metabolomics. So we've mostly had observational data, which means we take data that's already from example, like from a clinic, and the problem can sometimes be that you don't have the cohorts aren't matched based on, for example, gender or body mass index or age, which can be a problem because when we use statistical algorithms to detect the differences, you can actually be detecting BMI or age, not the actual biological effects of MECFS. And we've used some approaches such as this generalized additive model to reduce the influence of these potential confounders on our data set. And similarly, we've decided that the analysis will focus on sex-independent metabolic abnormalities. And the reason for this is that previously, I know a lot of studies have highlighted differences between females and males, but the problem can be that when it comes to treating these diseases, for example, multiple sclerosis, which predominantly affects females, there are not separate treatments if you're a male or female. So if this were prostate cancer and breast cancer, that would make sense because we know there's a strong hormonal association, but I don't think, to pursue this sex-dependent treatment makes much sense, really. And you can see here from our demographics table, the body mass index, it was different, so that needs to be corrected for. These are some plots here, just showing the ability of the statistical model to separate MECFS, which is actually green. So this is the unsupervised analysis, which means the computer doesn't know which group is MECFS or controls. And you can see overall, because there's a lot of overlap with green and the red, it's not able to separate it as well. But interestingly, the red, which is the healthy controls, they cluster very tightly. So their metabolome doesn't have much variation. They're very similar, whereas ME is very broad. And interestingly, when we go look at the supervised analysis over here on the right, that is able to separate the groups fairly well. But again, you could argue that you're introducing some bias because you're telling the computer this is MECFS and this is the other group, and hence where it separates better. But when it comes down to the overall statistics, it's actually a very robust model that's highly able to separate the groups, and overall the accuracy was 93%. So we need to do further validation with larger data sets. This is a plot here where you look at the p-value on the y-axis and the fold change on the x-axis. And you can see here that overall metabolites that are actually increased are here in red. They're up, it says there on the legend, up. And you've got the majority of metabolites here in MECFS are actually reduced, all these ones in blue. And this analysis just looks at the metabolites that we are able to identify. So in other words, there are some metabolites which we don't have a name for. Say, for example, amino acids, we can identify tryptophan, arginine, all these amino acids, but we still can't identify many. This is just a plot basically to show the importance. I know you can't read these very well of some of the amino acids, but yeah, the plots that you saw on the previous slides, you can see at the top here. So asparagine, lots of amino acids were quite important in discriminating MECFS compared to controls, and one of the glutamate metabolites also was pretty important, as was hypertaurine, which actually ties into is a precursor for bile acid formation, which is an area that has been flagged up in previous papers. And also, interestingly, cysteine, if you see here, that's the reduced form of cysteine, which means the oxidized form. So that's an indicator of increased oxidative stress. So that follows findings from many previous papers. And this is just a heat map, which also refers back to the top VIP ranked metabolites that we saw. And you can see overall, there's because visually there's fairly good separation, but it's not perfect, and you can see like potentially some outliers here which have abnormally strong profiles, so they need to be investigated more to see what's potentially causing that. And again, when we looked here, this was actually looking at random forest, which is a machine learning technique, and that was able to discriminate the group extremely well overall, with only, I think, one false positive. Thank you for listening, and that's the end.
Thank you, Jay. Any questions from here? Or? Yeah, Jamie's stuff's really hard to present. It's not easy. But a lot of information there. We're trying to write up a paper on graded exercise therapy where we don't see a difference. So Jamie didn't mention this, but so the controls and patients are different. We have half the cohort, this was done a long time ago now, so no one does this anymore, but half of them didn't want to do it or struggled and stopped. Half did it, 6 months, nothing changes. So all of those things that are different are still wrong. 6 months, so we're just writing up to try and get that out there. Because in long COVID, there's things going on in the US where they're thinking of doing.
But it's still being prescribed in Oxford. That's the problem.
Yeah, this is
Yeah.
absolutely. And this is what, just to summarise overall, what Karl was mentioning. Overall, we looked at around at least 2,000 metabolites, and these are the same subjects that's before and after graded exercise therapy. And if we look at these 2 time points, overall, there's no major statistical difference whatsoever, even if we go down to the level of like performing an individual t-test before and after on each value. So yeah, I think this is extremely important to be communicated.
My problem with that is that people who are being prescribed that aren't getting anything else. So I think they're being sold short. And that applies to a lot of people in Oxford.
What with long COVID?
Not so much long COVID, but at the fatigue clinic.
Yeah. Just
they're not offered anything else.
Graduate exercise at the fatigue clinic in Oxford.
Yeah. It doesn't work.
Nice, don't recommend it.
They call it different things now.
Right.
Well, you could try.
So could I just ask, so with the metabolites that looked like they were different in the ME group, was there anything, was it just oxidative stress, or were there any other clues?
No, there were lots of hopefully other clues. So one of them potentially is bioelastic metabolism, which I think has cropped up in papers in the past. So that looks like that may be abnormal. Oxidative stress as well. I think we looked at also I'm trying to think like glutamate as well.
Yeah. So glutamate shouldn't be in the blood. So glutamate levels are very high.
But it can be variable. So that's important to look at, because if the blood-brain barrier is permeable, then that could get into the brain, causing this excitotoxicity. Which manifests as hyperacusis, where people's hearing is horribly amplified and causing terrible suffering, and then could also affect light sensitivity potentially and pain.
The sad thing is, we have some things that are 40,000 times higher in the patients compared to controls, but we don't know what they are. So this is a real frustration of this type of work. Is we've probably got 12,000 things we detect, but we can only detect 600. So we struggle to get money, because what we need to do is we need to go again with another cohort, find these things that are different that we can't identify, and then work out what they are. And the way you do that is you can get so Edie showed you the chemical structures of things. So glucose has probably got 12 different things it could be based on the carbons and hydrogens and oxygen atoms. So then we have got to go in and work out, well, which one is it likely to be, and then make it. And so it's a huge task to do that, and it's beyond the charities. They haven't funded this work.
Is there more funding coming in with long COVID, one would hope?
Well, you'd hope it would be. There was quite a lot of funding on long COVID initially. But you've got to have the right applications. We don't really have many hypotheses in this area, not that I would put my hat on. So you're really competing. And the problem at the moment in the UK is that we have to compete in the big fund, the £2 million MRC grants, Wellcome Trust grants. We have to compete with all the other known established conditions, and this is the challenge. And we don't really stand a chance getting better, but things are moving slowly as we work out what's going on. So the money is there, but you need a good application. And when there's not a ring-fence pot, which there should be for these chronic conditions, and that's what we need to push government to make the MRC change their minds, and that's moving slowly. So there was going to be a talk on cryotherapy from Poland, but I've lost him. I don't know where he's gone. But it looks really quite an interesting method, and it seems to be giving some improvement for about 3 months. And we're just trying to work out why and what's going on. Cryotherapy in the UK will be more of a challenge. There are people in London that do cryotherapy as like a cosmetic.
Is that cold?
Yeah, minus 130 degrees. But compared to graded exercise treatment, the patients really liked it. You know, we had 50% dropout. They all came back. They did the whole course, and it showed some changes that we need to investigate. Okay, so I'm going to talk a bit about a blood test primarily, but also talk a bit about mitochondria, which I think is how I got to know Amiga through Norman Booth when he was got me interested in this area. So this is the group. So you've heard from a few Inga and Edie who are doing DPhils, and Jamie works with our big data. Does other medical statistics courses and a load of other things that he does. And then we have people that work on more fundamental areas of mitochondria. So Katie is developing methods, and Holly's just started looking at interesting stuff around how we make energy in a way that we shouldn't really make energy. I won't go into that today, that's another story. And then Megan works on some cancer therapeutics, which I think we could apply to some of these persistent pathogens, but we're really focusing on the cancer work at the moment. And then Fran is the first person we've had working on sort of Lyme disease. So she's got some samples. She's only a day and a half a week. We share her with Brooks. It's going a bit slowly, but we're making progress. So we are sort of quite active. And these things are sort of all come together at some point. We mustn't forget Janet. Janet always wants to retire. And I keep saying, I can't retire because she's so good at imaging. And I'll show you some of her data during my talk. So that's the sort of group. So one of the things we really need to do, not just us, but everybody, all of these areas, autoimmunity, gut dysbiosis, sort of metabolic disturbances, which Jamie mentioned, energy metabolism, mainly at blood cells, but it goes probably further than that, autonomic dysfunction, and immune cell issues are all things that are there, but where are they sitting in the condition? Which is one of these the driver? Which one of these should be hit to try and stop the illness? Or are they just consequences of being ill with something else that we don't really know about? And that's where we need to go. We need to try and move from associations, which is what most studies generate when you've got a control group and a healthy and a patient group, to causality. And that's the big challenge. It's something we need to sort of try and work towards. So in general, we work on ME/CFS. We've done that for about 5 years. We just started with Inga's work to look at long COVID, and we're going to be recruiting patients to the study. We have some samples from a Dublin clinic who work on chronic Lyme disease with antibiotic treatments. So we're trying to work out what's changing when patients, because 70% of patients with chronic Lyme do really well with antibiotics. 30% don't, so why not? What's the difference? You can work that out. And these other conditions are all chronic. Pandas is another one. Endometriosis, we don't really know what causes them. So we don't work on these, but in our department, there's a huge group that work on endometriosis. So there's lots of common ground between the groups in this women's health unit. So if you look at long COVID and ME/CFS, they're very similar, apart from a couple of things. So the smell, the taste, the chemical sensitivities down the bottom. But if you think about long COVID, it's quite a new illness. We didn't have COVID-19, and so ME/CFS, if it's triggered by something like Epstein-Barr or other viruses, it's been around for thousands of years, so it's probably evolved to this point. So they're really similar, and that's helped a little bit in what people think about ME/CFS, and I think they're starting to, the conception of how they treat it is different. You don't need me to tell you this. It's a real problem. Many people affected, and I think Edie has already picked up on this before. So this is an interesting system. So the James Lind Alliance, many of you are probably aware of it, is the patient groups get together and they work out what are their top 10 things that they really want to understand, us to work on as researchers. So the top one is post-exertional malaise, which has been mentioned, and we may get some handle on that with Edie's cuff. Jamie's studying graded exercise cohort, so we'll see what changes there. Exploring existing drugs, what drugs are out there that could be useful, and that's a challenge. Lots of people are talking about low dose naltrexone, various other compounds. How do we study those in a clinical trial? Because that's what we need to do. How do we pick the patients? And that's the challenge. And the third one is to develop a test. You know, there's no test. You go to the doctor, they come back, everything's fine, nothing wrong with you, they go away. So can we find something that can really label a person as, I mean, an early, you know, if you could diagnose someone within the first 6 months, then maybe that might change the whole outcome. Maybe they will do the right things, and so that could be really important. So I'm going to talk about a method, Raman spectroscopy, which Edie has also mentioned. So we studied it at a single cell level. There are a few ways to look at what's going on in a single cell. So you can have fluorescent tags that tag structures in the cell. You can do sequencing of the RNA, so all of the sort of chemical fingerprint of the cell at the DNA RNA level. You can look at that. But Raman spectroscopy could give you a handle on what the cell is actually doing, and what is it doing in real time on a single cell or even a tissue. So that's what we're trying to use this for, and Edie's talked a bit about it in her HUVEC model, but what I'm going to talk about in this talk is it as a diagnostic tool. So Edie's already mentioned this slide, so you hit material with a laser, you get this vibrational energy that you can pick up. It's really weak, so we do have problems with fluorescence in samples, but we can get around some of that. I won't go into this. It's some new microscope. It's got lots of mirrors, lasers, various things in it. So it's quite expensive. I think we're just trying to get one. It's about £250,000. So we're trying to get one for here, which would be fantastic. And as Edie said, you get a fingerprint. So it's a fingerprint of the cell, what the cell is actually doing in terms of things it makes over here. So you have all these. Just go back to So there's regions here. So the biological material will come up in this region and also over here. This is called a silent region where things shouldn't be, but we do pick up some things here. So heavy metals will be in this region in the middle here. So you're trying to use this as a fingerprint. So, you know, we want to see whether we got a healthy fingerprint for a healthy person, and can we find a disease fingerprint from these signatures on the single cells that could help us. Now, they're going to be really complicated, and I'll show you in a second. It's not going to be one thing. Like you go and if you have a heart attack, you have a couple of molecules that they can pick up, or your liver's failing, 2 molecules. This isn't like that. And one of the challenges is going to be, if we find this and it works, how do we persuade the doctors to use this as a tool? Because they're not going to, they will say, well, what's different? And I don't know. It's going to be the whole picture, but it'll happen. So we have to take these fingerprints and use complex machine learning methods, sort of AI methods, to try and work out what's different, and I'll touch on that. And then we want to come up with a diagnosis. That's the ultimate goal. So somebody asked the question, has this been used before? And yes, it has. So this is an example of early stage lung cancer. So here they're looking at things called exosomes, which are tiny round structures that are shed by cells into the blood. You can isolate them and you can look at them. They do that by Raman. You get these peak profiles you can see here, and they can tell that the ones from the cancer cells are very different from the normal cells. And using this, they can pick up a fairly early stage lung cancer diagnostic test, and obviously this is something that they're pursuing. So in cancer, it looks useful. Again, machine learning. You can't do this by eye. It's got to be done by a machine, a computer, to do the analysis. This is quite exciting. So, you know, if you have a tumor, and sometimes they're in places that you can't really get at, Raman can see the difference between a normal cell and a cancer cell. So in a way, you can be guided by the Raman laser, and I've seen a machine that does this, and it's like a big thing that you shine on material, and it can find the margin. So you could find the margin really easily, and then just dissect away the cancer. It will take a long, it's quite slow at the moment to do it, but in theory, you could get, so in the brain, for example, you could get really close to the brain tissue and take the whole tumor away and not leave any behind, or the spinal cord. So this could transform sort of surgical methods. So it's been used, and it's an evolving tool. So this is a summary of what we've done. So we've taken healthy people. We've taken these PBMCs, which are components of blood, the white cells, the immune cells. So there's T cells, lots of components. It's a mixture of stuff. So it's not just got one type of cell. We also have done this for the multiple ME-CFS group, same thing, PBMCs. And what we did, which is quite different from other people's studies, rather than just say, let's look at health as the controls, we wanted to use a control group that was ill. So we chose MS because it's quite similar. There's a clear diagnostic test for that with the MRI. And so we had another group that, so we don't just want to know that our patients are different controls, because what does that mean? But if it's different from another group, it becomes more likely to be potentially something really useful. So we look at the single cells on the machine, we get to profiles, and then we see whether we can fingerprint the samples. Now, can we tell the diseases apart? But the other thing that we did, which is really interesting, can we tell a severe patient apart from a moderate one or a mild one? And if we can, that'd be really useful. And coming over here, so, you'd come in for a diagnosis, you do the Raman, you do the machine learning, and end up. So this is the ultimate goal. You want to have a blood-based method that can diagnose a condition. And if it can tell you whether you're mild, moderate, and severe, but you probably already know that, it could be quite helpful as well when the people drift around. So we got samples from the UK ME CFS Biobank, which is the ME Association Fund, also the NIH. Doesn't get any funding from the UK government, which is quite surprising. If anyone's listening from the NIHR, then that's useful. 98 subjects, 61 with the ME CFS group, 21 MS, and Jamie mentioned this a little bit in his talk. We lost 4 of our healthy controls because they had BMIs that were too high for our study, so we couldn't incorporate them into the study. So we have to be really careful who we select, I think, for these studies and get that right at the beginning rather than find out halfway through. Because we got these studies blinded, and if I did this again, I wouldn't want blinded, because nobody really trusts the scientists, unfortunately, in this illness. Everybody wanted us to work on blinded samples, but really we really needed to know that they were good before, because we weren't preparing them. But with Inga's study, we will get the right people and not have problems like this. So the first thing we did was we looked at the mitochondrial function using standard assays, a bit like the assays that Norman Booth developed with Sarah Myhill and Acumen test. And we didn't really see a difference. So using this test, we didn't really see much of a difference. And we think this is partly because these are frozen samples. They've been frozen for quite a long time, and that impacts on this assay big time. And I've now come across groups in autism. In autism, you also find mitochondrial dysfunction in these blood cells. So it's not unique to this condition. This is the cohort that we looked at. You can't read this very well. You can look at the paper, it's out. So these are different components of the illness. And if you've got a very high red signal, it means you're really quite severe. So the severe patients are over here. You've got the moderates and the milds. This is the MS cohort. So you can see they're really similar. Lots of similar symptoms to the ME CFS group, particularly the mild, and as you get more severe, they get worse. Healthy controls, and clearly even in that group, there's things that are similar across the groups. And so quite a nice clinical data set. 98 is quite a good size of study. And then this is what we got. So this is a complicated slide. So you can see here, these are the spectra, the average spectra of the different groups. So the green is the healthy, the red is the ME, and the MS is the blue ones. And by eye, you can tell me which one's which. I don't think you could very easily. This is the mean of thousands of different spectra, and they're quite tight, which is always quite surprising. So there's not a big variation here. When we do this quite simple machine learning method called linear discriminatory analysis, which takes probably a component of this spectra and plots it in a sort of a linear, takes a complex data set and picks up features, they separate out really nicely. Now, they're not significant at this point. They're just really different. So this was quite nice to see that we could actually separate the groups, 3 groups, really quite nicely. And then we went on and added different complexities. So here what we've done is we've added the different severity groups. So you've got the healthy control again, the mild, moderates, and severe. And what's interesting here is that the milds separate out from the other 2 groups. So the moderates and severes overlap. And the healthy control is also, again, really different. So that was good to see. Interesting that the milds were different from the moderates and severes, and maybe they're very more similar. And then we added in the MS group here. So there's 5 components. And so what we do is we ask the model to say, can you separate them out clearly on the spectra? And this was a little bit different. So here the control groups always come out as different. In this time, the severe groups stand out on their own. And the MS group, the moderates and milds are all stacked on top of each other. So depending on how many groups you look at, you get different answers, but always you can separate the control group out from all the others. And this is an interesting plot here. This is a plot of the severe ME patients against the healthy controls and the MS patients against the healthy controls. You can see there's clear difference in the profiles at different parts of the spectrum. So there really are some big differences there. And there's more on this in the paper. So that looked interesting, potentially something we could do more with. So just to take it through a bit more, so this is the protocol. We take a blood sample, we purify these cells, which band here in a gradient. We stick them on the Raman system, so this is the cell, hit it with a laser, and then we take probably 5 points in that cell, and then we produce a spectra, and then we do that, produce an average spectra for a cell, and then we do 30 cells per patient. So we have, I think, about 21,000 spectra. So it's quite a lot of data, and it's all done quite manually, which is quite tough. Edie would say that that's tricky to do. We hope to get it automated. And then what we did in the end was we looked at different methods to look at the data. So rather than just using one machine learning method, we use 5 in this case. All of them individually are slightly better than chance at pulling the groups apart. But when you use the power of all of them, we get a really quite high diagnostic potential. So we have a trainer set, and I'll come back to this in a minute, and a test set. And then we run them through this ensemble learning method, and we try and see whether we can group them. And so this is, because the sample, even though 98 sounds a lot, it's still quite small. So we have to run a training set, which Edie said was 80% of the data, and then we hold back 20, and we use that 20 and say, can it predict where they go? But we did this 5 times. So this first one, we took all the data, allocated some as the training set, test set, and then we did it again, and we picked out a different group and picked out a different group. So it's not perfect. Ideally, you want to have a training set, a test set, and then a group that you bring in that you don't never touch before. It's the final thing. So that's what we'll do in the future. And then based on this type of approach, we then see whether we can pick things out. And so what was really nice was that we were really quite good at picking them out. So 90% of the time, we picked out the MS ones correctly from the others, from one of the sort of test samples, which wasn't in the model. And then when we looked at the severe, moderate, and mild, again, we could pick them out quite well. So this is quite exciting and something we need to develop as a take forward, because now we have no tests. So this looks like it could be useful. And so this is our future plan. So this is just an explanation of what we got here. The other thing you can do with this Raman method is you can use other chemicals in there to help us really look at what's going on. I won't go into details, but it could be really quite powerful. And then maybe combine with other things like wearable devices to try and produce this sort of big data groups, like on Inga's project, and see whether we can mine things together and come up with some really useful sort of methods. So the next step, we're just talking to the ME Association, and hopefully this will be sorted out. So we want to run another validation study, slightly bigger. We are limited with resources. We can get the samples quite nicely from the biobank. So 40. We're going to stick with mild and moderates because the severes look quite different. So we're going to leave those for now compared to the multiple sclerosis. And then we have bigger training sets, a test set, and 5 we've never tested. And hopefully that will work well. So we're going to work with 3 other centres, one in Limerick, which is a Raman group, and Alan Moreau, who's the chap in Canada that's also interested in this work. So we'll prepare the samples, we'll send them the grids, they'll do their analysis, we'll do ours, and we'll see how we comply. So that'll be really cool. And we think that'll take about 4 to 5 months. And Megan, who's currently on the cancer work, can use Raman. She should have finished her DPhil by then. So you start using the people that you've got in the group to do that. So that's quite exciting. So that's the diagnostic. I thought I'd just mention a few more things. So we started off studying mitochondria in ME/CFS. It was really difficult to do it on the blood cells. You saw from that previous slide, when you freeze them, they're not easy to work with. Fresh ones, you've got a fresh sample of blood. It's really hard to work with a fresh sample. There aren't very many NHS tests that work on, they work on fresh samples, but they've got a really good validated cohort. Lots of evidence that mitochondria are impaired. So Norman stuff sort of did this 5 or 6 years ago, other groups. So we really know that there is a problem with energy metabolism in these patients. So how do we study it? And this is an assay that we do in the lab. So 2 cell lines, you don't need to know really what they are. One's got really high mitochondrial activity, and one's a bit like a patient. So low mitochondrial activity, and you can see the A549 are all down here. The H460 is up high, and we can do things to the cells that make them go faster or slower. And so we've got an interesting system here where we've got good and bad mitochondria, and can we study them? So what we did is we took the mitochondria, see a little green spot here, that's an isolated mitochondria with a Raman laser shooting it. So you can see the green. So we do that on quite a few samples, and we produce the spectra like I showed you before. So this is a bit complicated. So this is a very mitochondrial active cell line. We've basically given it some food here in this one. We also hit this one, we've given it some food plus a molecule that will make energy, this ATP you might have heard of. And we poisoned them in 2 ways with different approaches. And what you can see, this is complicated slide. If you look at this, so this is the food on its own with no ability to make energy, and we see a big change here in that one. And when we add the poisons, we wipe that out. So this is something interesting. And then, but not just there, there's over here as well. So this is the food. We add the ADP to make the energy. And again, there's a big shift this way. And again, when we add the poisons, we wipe it out. And then the same in the middle. I'm not going to go through that. So here you can see again, there's a big change with this peak. And then you restore it again. So we really believe that we can actually study mitochondria inside cells in different systems that are good or bad at making energy. Now, blood cells are terrible for this. Blood cells have got a big nucleus, hardly any cytoplasm. You're not going to do this on a blood cell, but you could do it on other cells. Edie can do it on the HUVEC model when we add the serum, and we should be able to track mitochondria in her HUVEC cells, which are really big and got beautiful mitochondrial structures. But what about this type of system? So as I showed you, these are mitochondria staying with the dye, and some cells from a patient could have things like the muscle cells. And so what you do is you come in with your laser and you'd interrogate these different parts of the cell and do that. We could do it. We just put in a grant to try and get a new microscope. At the moment, we use one in engineering. It's 15 years old, gives terrible images. This one's £250,000. We just put in. I'm hopeful we'll get the money. It'll be here because there isn't money in medicine. So this will be a first for medical sciences. We had to play with one last week down in High Wycombe, and we took some retinal sections, and it was amazing. We could see the rods and cones just on their profiles of the Raman. It didn't stain with anything. It was really quite remarkable. So hopefully we'll be successful and we can do more. Now, we could look at muscle. So we could take a biopsy from a patient. This is a mitochondrial disease. So you see these red stains here. That basically means that the mitochondria are failing and you've got a real increase in mitochondria, but they don't work very well. So this is a really severely ill patient. People have tried to do this in ME. I think there was a group in Glasgow did this years ago. You see a few things, but I don't know whether you'd see this type of difference with this staining. But the Raman could be more sensitive. However, you've got to get healthy controls. So how are you going to get healthy controls to donate? Muscle biopsies are quite invasive, and I think it'd be difficult to do. There is a paper that came out of America recently that shows mitochondrial problems. But the other question is, where does the mitochondrial damage fit in the illness? Is it something that's here and something else is doing it? So you might be able to prop them up a bit, but is it really going to help? You know, people try different vitamins that could boost mitochondria, but do they work? So taking a biopsy off somebody is going to be a challenge, but we can do this in other ways. And so there's another group, they're not here, they're away. Ladislav looking after his kids because it's half term, and I think Beata's off on a conference. So they've been studying mitochondria and ME and CFS patients and long COVID using imaging. So they use MRI imaging. So you've got Beata's based in psychiatry, but she's more a molecular person. And Ladislav works in RDM, which is like cardiovascular. So he's a heart person. So Beata looks at the brain. So she's been doing this 7 Tesla imaging. So you can look at what's going on in the brain. And like Jamie showed, you can get a readout of the chemicals. If you go in for a brain scan, MRI scan, they look at the structures. This is another level up, and you can look at the compounds in the brain. So you can see things like glutamate, GABA, the different transmitters, glucose in the brain. So this is quite a powerful method. So what they're doing is they're almost to the end of the study. I think it's getting quite close. So they're looking at 25 ME/CFS patients, long COVID, got the controls. They go through questionnaires, but like Inga, and they do a challenge for the brain. And also, they do an interesting thing for the muscle. So you can take a patient, put them in a magnet, and they do a mild bit of exercise. And this is really cool. So this is the energy producing muscle. So you've got the ATP that I mentioned before, ADP. But in muscle, it's the phosphocreatine that's really important. So if you look at this bottom graph here, so this is a rest. So when you do a scan across the muscle, you have quite a lot of phosphocreatine, big peak, and then you exercise and it goes down. And then when they exercise, they rest again, the phosphocreatine goes up. So this is an example of one of the patients. So less phosphocreatine at baseline, they go down and they don't really recover very much. But if you look at the ATP, that can be quite solid. So this is that graphically shown. So the phosphocreatine is the blue. So you come into the scanner, you start exercising, and the phosphocreatine drops down. Other things change, so that the PI changes if it's a component which fits with this going down. ATP stays pretty solid, doesn't really change very much in this individual. And then there's a change here as well. So this allows us to do things in patients, not taking a chunk of muscle. So you can actually study what we need to study in a real patient. You could look at the brain, you could also look at the liver, so you can look at any aspects. It's quite an expensive method. We have a scanner in Oxford, and if we do run a big clinical study, we'll use it in that study. So that's me. I thought that's to give you a whistle-top tour. So we have most of our money. ME Association have kept us alive, essentially, for 5 or 6 years. Amiga, you gave us money. You gave us a donation years ago, and that's been really helpful. We recently got some money from Action for Me to support Inga's work, and the Lyme Treatment Foundation supported the Lyme stuff and various other things. The Dove Clinic, we worked a bit with the Dove Clinic. Sadly, they shut now. Julian Kenyon stopped that. He's retired. We've got some really exciting fecal transplant data in collaboration with this company, but unfortunately during COVID, it all got lost in the freezer and all the soft cell change. So Julian's been doing fecal transplants on ME patients for years, and he says, oh, it works. They don't come back. When I say to Jamie, well, they don't come back because it doesn't work. And so you've got the doctor saying it works, and then patients are a little bit more skeptical. And I said, well, why don't you work with this company, Soft Cell, that are looking for bacteria in samples in blood. And we did one patient, and it looked really exciting, but we've only got an N=1. But I really think the pathogens are really important in this illness, and it's just working out what they are. And I'll stop and I'll answer any questions if there are any questions. What type of mitochondrial function in lay terms are you seeing? Yeah. So this is the paper I mentioned. Thanks, Sarah. So it's hard to know. So a lot of people use a system that's called a seahorse. It looks like a seahorse. It's got a shape of a seahorse, but it measures the ability of cells to use oxygen. So mitochondria use oxygen. So if they're good, they will use a lot of oxygen, and you can measure that output. It works on blood cells, which I don't like very much. It's really quite hard to get to work. So they are showing a deficiency in the ability of the cells to use oxygen. Now, it could be the mitochondria, but if you remember my analogy with the food, we have to add the food to the system. The food starts up here, so your glucose. It goes through another pathway before it gets into the mitochondria, and then it feeds in. So if there's a problem with mitochondria, then you may not, they can't see this on there, can they? So you may not have reduced oxygen, but if there's a block up here, it could also affect that thing. So it's not clear to me whether there really is a mitochondrial problem or not. But there is some evidence suggesting there might be. But it's not a catastrophic one like the patients. It's something is turning it off. And people fluctuate. Some people, cyclical timers, go up and down. Why do you go into post-insertional malaise? What's going on? Is it the mitochondria? Are you working with, I have this theory about, are you working with pathogens? Is there an interaction there? So boosting mitochondria might help. But how do you do that? It's not clear. Okay, someone can't hear me. Okay, I think that's it. Cool. So that's us. We're trying, and we're doing quite well. I think we're getting to the point where we might be able to apply for some bigger grants. We've had a couple of goes and got bounced by the funders. These big grants, they're hard work. You spend probably 2 months writing it, and then you get it bounced. It's not much fun, but you have to get up and have another go at some point. But we've got to be strategic. We've got to work out how to do it.
So the charges don't just bounce things back, or they don't?
No, we put in a good project on Lyme disease with Jack Lambert, antibiotic. I thought it was really good, and they come back with things like, we don't think the group have got enough experience. They didn't like the treatment. Just 2 lines. That was 2 lines. The other ones, MRC ones, the problem is you get people on the panel that don't believe this is a real illness. So they will come back and give you our first ever grant to the MRC. You get a score out of 6. We had 2 6s, a 5, a 4, and then 2 1s.
Right.
And then when they only fund 18% of the grants, you're gone. So this is one of the big discussions with the MRC at the moment is there's no point. If you want to fund this research, you have a panel that believes it's real, or people on the panel that want to fund it, want to fund the biological research and don't kick it out, because and I'm not going to put another grant in until they change it. It's not worth it.
What about the link with the James Lind? There's no kind of they've got these questions. They say, oh, these are the research. Make sure if you're applying for funding that they answer what James Lind. Yeah,
we do that on the applications, but I think MECFS has been an MRC highlight notice for at least 10 years. Apart from the big Decode project, which I think they just fed up. They got fed up with the people nagging them to fund it a little bit, and who can do something on that scale in the UK? There isn't really people there aren't the researchers to apply. We're probably one of the only groups in the UK that there aren't very many, because you don't last very long. So,
It does seem to me it's a bit of an Oxford problem specifically. So I moved from London 5 years ago and really hit a brick wall in Oxford, and it seems to me
the treatment.
Yeah. No, it seems to me if you've got a group of people who don't believe it's a real disease and you're trying to get funding, those two are not going to mix.
It's the same with any area of science. You think about, there's this guy, Max Planck, people have got to die before people change their mind. And you think that was something in the 1800s, but it's probably as true now. And Alzheimer's, we've worked on amyloid for years. The evidence is really weak. It's probably an association, not a cause. So all the drug companies spent billions and billions of pounds on that, and it hasn't really gone anywhere. And they've squashed all the grants that have come in on other areas, which is still there. Infection's been a big component of that, but no one's really studied it because the grants have never got off the ground.
Yeah.
So is MRC the main source of funding if you really wanted to?
At the moment, yeah, probably you can go to in this country, the MRC, you have the Wellcome, but they've changed their remit. They tend to fund tropical medicines so much now. It's a shame because they were a really good funder. They have these there's a big horizon projects where they fund like 7 years of research. But you've got to get to that point to be competitive with that. You can't just go in. So slowly, we have to work slowly, I think. You might get a big donor. In America, there's some big donors that donate money. There's Open Medicine Foundation, you might have heard of. They've had about £40 million. But it's spread around lots of areas. I don't think there's a big pot of money over there either. The NIH has funded 3 or 4 centres to $20 million.
It felt like there's no shortage of, if you had 100 people and 3 Ramans, you'd have no shortage of ideas of what to look into. Or
the Raman's an interesting tool. There's a new group coming from London, from King's, down into Molly Stevens, down to DPAG, down the road in biochemistry. She's bringing 100 people with her. She's a Raman expert, so we're going to have a really good critical mass for people doing Raman. So she does lots. It has a few challenges because it's quite weak. Edie will see cosmic rays on her screen, and when somebody says, "Oh, it's a cosmic ray," and you think, "How does it measure a cosmic ray?" And then it will go out the other side, and then you look at it again, it's gone. So, and I was actually down at this company, and they said, "Oh, well, we get cosmic rays coming up." So any colour can really influence it, sort of fluorescence, even mildly fluorescence. So you've got to work out. We can use it for cells, but using it for tissue is going to be a challenge.
Okay, well, thank you so much, Karl,
yeah.
and to your whole team, to these guys at the back here who soldier on, doing wonderful work for us, because and for our members. And, if anybody's got a spare $250,000, want to spend it on a Raman spectrometer.
Father Christmas.
Father Christmas.
Yeah.
It's an Oxford scheme. I'm quite confident. I didn't know it existed, this scheme. Hid it away down in town. The funder.
But I mean, seriously, if we can get more fundraising off the ground amongst Omega, I'm not sure how we can do that, but if we have any experts out there who are watching online, you can see me, on fundraising and things, we would love to
get the MPs to help. Yeah, the government, I think Savage Javik put this thing together with, that's moving, but it really needs the government to tell the MRC they need to ring fence the money, because they won't do it otherwise.
They're doing a consultation at the moment.
Yeah, so they need to push that. They need to ring fence the money for this. It needs to come from the government, because they don't want to ring fence it because they say, well, that's bias. because you're biasing to that illness. And they've not done it for ages. I don't think they'll change. The NIHR were talking about ring fencing money, but there'll be clinical projects, which will be useful.
Well, lastly, thank you all for coming, the people who are present and the people online, and for taking interest in this. And thank you.
Thank you too.